Okay, I think we should get started for our part 2 for the interest of time. So welcome back. I hope you guys had a little refreshing break just now. So we will start from our first question. So the first question is, when population pharmacokinetic models are used for generating model integrated evidence for bioequivalence, what would be the appropriate model validation strategy? And what are the basic elements for model integrated evidence to be considered suitable to demonstrate bioequivalence? So I want to elaborate a little background for this question, for the thinking behind this question. So for pop PK to be used to describe the PK and for other purpose in the new drug development, most of the times the pop PK are more validated based on those predictive checks and some other more like, I mean, you may can call them qualitative checks. But for the BE purpose, because we really want to, I mean, for this purpose, and we're thinking maybe you need to do something a little bit more. And as you heard from Andy's presentation this morning, he actually shared with you, like, you may really want to compare the mean and the distribution of the Cmax and the AUC from the simulated data and observed data. So that's something actually different. It's more like so-called quantitative validation on the pop PK model itself. So I really want to open our first question on the validation in your mind, that what would be the appropriate model validation strategy. So I will stop here and I will turn it over to you, panel members. Okay, Murray, go to you first.
I can start. I mean, there are two different questions in there. I guess I'll focus on the first one. First of all, we don't want to reinvent the wheel. So we know from decades of experience on how to validate a model internally and how to validate it externally. It's just that it was not for, let's say, a generic product, but there's a lot of knowledge that we don't need to reinvent and that we'll be able to use. At the minimum, if you have a perfect population PK model to start with from the literature or from other sources, then at the minimum, you will need to have an internal validation. And the internal validation, depending on what you want, on what you're doing in terms of your model BE, will be different. We touched a bit about that in our paper from 2 years ago. And if I take something that Dr. Zhao mentioned earlier, that the validation has to be for purpose. So depending if it's a virtual BE, the internal validation will need to be much more stringent than if it's a continuation and you're not inventing new patients. That would be the minimum. If the pop PK that you start with, that you use to come up with your innovative study design and that you prove that you control the alpha error and the power, etc. If that model is not perfect, if it has some limitations in it, then I would see that you will need, in addition, an external validation or something to improve that model and make it okay for the model-based BE. So I would see a pilot study at the minimum need to be done before the pivotal model-based clinical study design is done.
Could I chip in there and just ask? So you said if a model is publicly available, it might not be perfect, but you need something in order to at least get to a pilot study. And I think this is what Joga and Andy also mentioned in the first part of this. That you need something in order to build a profile, and which you can then validate using a pilot study. Because Andy's point was you can't use your data from later on to build that, because you're biasing that. So you need a starting point, and that starting point has to come from the reference product. But if only 8 out of the 30 that have been approved have a model that is publicly available, what about the other 22? And what about new products coming to the market? What is then the incentive if that model that they make publicly available is then going to be used for a generic later down the line? What is the incentive to then make a POP-PK model or a PBPK model publicly available?
Can I follow up with a clarification question just to make sure I fully understand? So what is the purpose of the pilot study? Is the pilot study mainly for validation? That was, Mira, you mentioned the external validation. You are not necessarily going to include the data from the pilot study in the building of the model. Is that the value of the pilot study, or you are also talking about we are going to build the model using the data from the pilot study? I just want to make that point clear before we move further.
So what I think what everybody said is you have to take something from the reference product and use the pilot study to validate that model or refine. And Murray's point was that model from the reference might not be perfect. You might need to refine it and use the pilot study to build a refined model of your reference product. And then you can use that to then test your own formulation in your own PE study. My question goes before that. So to get to that stage where you're either validating the reference model or validating a refined reference model, you need a reference model that's publicly available. If that model is not publicly available, as in 2/3 of the long-acting injectables that are currently on the market in the US, 2/3 of them do not have a POP-PK model or a PBPK model that's publicly available. How can any of this approaches work? That's my question.
Yeah.
Okay.
Can I answer? Parmesh, I mean, many times the published PK model that is published or available, we see flaws in it. There's things missing, even when it's published. And if it's not published, then in all of the situations that we've seen before, there is enough PK data in the literature that you can come up with what should be a structure of a population PK model, and you can come up with something that is reasonable to do your initial simulations of your study design that you want to propose, the alpha error assessment and all of that. But in those two cases, then to answer Lucy's question, then I think a pilot is needed, because you want to make sure that the model that you've used to justify your innovative study design, to justify the control of the alpha error, you want to make sure that it was appropriate. And that pilot data will be able to do that. In the best cases, you will be able to validate externally what you've done. Or if it doesn't validate, then you will have to use that pilot data to come up with a better population PK model.
Okay, that's clear. Thank you. That's it.
Lucy, I want to make it clear though. Your question is referring to the population PK model, not the simple
Correct.
PK model, like what is 1 compartment, 2 compartment, or 3 compartment. There's a difference between the two. I agree that there are many literature information on PK models for different drugs, but in terms of population PK model, I don't think that's widely published. I think as Parmesh illustrated earlier, that most of the LAI do not have the population PK model published, and to me, what's important here is the validation of the population PK model, not the validation of simple PK models. And we know for the new drug side, validation of population PK models most of the time are probably done with visual comparison between the predicted data and the observed data. They don't have very stringent criteria, unlike the generic side where we have plus or minus 20% on the AUC or Cmax. They may not have that kind of stringent criteria. So question is whether that kind of validation is adequate for BE determination. I think that we need to be clear on that.
Anyone wants to? Okay, Andy, please.
Well, maybe we can let Murray respond, I think, and then I can hop in.
Yeah,
yes,
just quickly.
please.
Yeah, just quickly. It's basically you Chang. It's again going back to Dr. Zhao was mentioning. Like validation for purpose. It depends what you want to do. If you want to do a virtual BE, then the population model is immensely important. I mean, that's what you're gonna be using for simulating new patients. But if you only are continuing patients that you actually dose, okay, there's not gonna be new patients, then what is important is the structure of the model, much more than the population, because you're gonna be using the individual PK parameters of all those patients that you're continuing. So then you just have to validate really the structure.
Yeah. So what you're saying for in silico simulation, perhaps we may not need as stringent validation on the population PK model as if you use it for virtual BE determination. I think that's what the point is, right, Murray?
Yes, that is correct. Yes, that's correct.
In Murray's case, because you have individual data for like at least following single dose for test reference, then you can just project what would be the steady state PK profile for that individual patient. Then for the trial simulation using a virtual population, I think it need additional layer of validation in terms of whether the PK intra-subject variability has been sufficiently differentiated and characterized. In that case, I think Andrew is kind of has a long standing in using different approaches in terms of doing model diagnostics. We use different terminologies, right? Model evaluation, model diagnostics, model validation, assessment, and then for the visual predictive check, just if we just have a descriptive description of PK, then it's okay. Let's use VPC. But if we want to really understand whether the uncertainty has been sufficiently characterized, we may use numerical predictive check, and we also want to use some other like a type of validation techniques, like MPDE and other SVPC, from another industry group from Pfizer, and then if we feel comfortable, the simulated quantiles always is consistent with the observed quantiles, then we do have a confidence in terms of we are not only evaluating the center tendency, but we are also evaluating peel off at different layers of uncertainties. If we have a good confidence on those, uncertainty evaluation from different angles, then that could be good. That can make the modeler comfortable, and even non-modelers comfortable to see, okay, the simulated world is kind of have a fingerprint matching with the real world. But having said that, we many times may not need that stringent evaluation that could implicate a super large sample size, for example, to allow you to have very good estimation of a quantile from observed population. Then you need lots of patients. In that regard, we may need there could be some technical breakthrough that need other alternative approaches that need the field to work on. The game is not over yet, but certainly depending on the risk of the product as well. So I think FDA is always talking about a regulatory consistency, but sometimes when the science is evolving, the consistency part could be very hard to control. Sometimes we may need like an expert like us here. That do we all feel comfortable to trust that model? And given all the integrated information, the in vitro testing I see in the comment, there are Q1, Q2, Q3 testings, dissolution testing, the stability testings. On top of that, we have model integrated approach. Then, given everything together, we may need, for some cases, we may need less stringent model validation. If more sufficient, stringent model validation lead to unreasonable large sample size, then that could be something else we can think about, by using weight of evidence approach on those. We cannot always criticize the model. Have your model be sufficiently validated? And there are lots of other factors in the equation we may also need to take into account.
Thank you, Liang. So, Andy, I see you want to make a comment.
Yeah, I was just thinking about Parmesh's initial thought. So, and I think it's related to Liang's comments as well. If there's absolutely no population model that you can derive from any prior information, then I might suggest that at least one way you might go about building a population model is somehow either building your model based on only the reference data from a BE study, because really what you're interested in is the difference between the reference and test. So it's that difference you're trying to get at. So if you build your model just based on the reference, then you can use the rest of the data to investigate where the test is different than the reference. The other thing you might think about doing is building your model based on all the data, but blinding yourself to whether it's test or reference data, and then doing a bioequivalence test based on adding in those test components to the expected, to the absorption parameters, and then testing whether the size of those effects. I think both of those approaches might be ways that you could then tease out the population model without biasing yourself too much towards the difference between test and reference.
Andy, in that case, would you have to do the full validation of that model that you built in either case? Either in the case that you've just taken the test or you've taken both, but you're going to have to do a full validation in that case of that model to show that, as Liang said, all of the visual predictive checks, consider all of the residuals to make sure that that model does actually represent your data.
I would guess so, yes. But I don't know what that, yeah. So yeah, you would probably have to then go through a battery of evaluations of your model to see how well it's fitting. I agree.
Hi, Lucy. I have a few comments. I still don't understand why we keep going back. Maybe my microphone is really not working. Um, we don't I don't need a published model to start with. I just completely don't see the requirement for the brand company to publish a model for the reference product for two reasons. One, scientifically, I don't need it for developing a generic, and two, it's an undue burden on the generic company. So that's for both those reasons, I don't need it. I don't think we should expect the generic company to find one from the published literature. Second point is for this question, the focus here is my proposal is that we need to characterize we need to use the model to characterize the pharmacokinetic profiles of the individuals who for both test and reference products from this abbreviated. I also discourage us from using this pilot study. I am not referring to a pilot study. I'm talking about an abbreviated BE study, meaning this is serious business. You will have a plan and you will discuss that with this. Pilot study, you will have to do a pilot study before the abbreviated because you don't know how the in vivo characteristics of this quotes test formulation you're trying to move forward, right? So that is a pilot study. Maybe you will have to screen 2 or 3 formulations before you pick the one that you would like to move to the abbreviated. So can you combine the two? Sure, you can. But I want to call this abbreviated, not pilot, meaning this is serious. So in that case, that we need to the design of this abbreviated BE study should be robust, meaning you should design it for success, meaning to describe the profiles of each patient. So that way I have I'm using the model as an interpolation. So once I have confidence that the model describes the observed concentrations well, for a well-designed study, I can come up with a metric saying that my prediction should be within a much of the observed concentrations, and then I use that to calculate the PK parameters of interest. Then I can perform simulations even either using a parametric or non-parametric approach at that point. So the focus here, the validation approach, should be based upon the ability for the model to describe the full PK profile for both test and reference accurately. Now, why do I care if the model for the reference and test are different? I don't. We should not. So the reason is, let us say they are different shapes. That itself is going to tell you there's some difference between them that can be used towards the regulatory review. But even if they are different, my focus is still on the PK parameters of interest, the partial AUCs, the AUC Cmax, or any other parameter that I know of is of importance. That has to come from a regulatory perspective, period. So because that endpoints are selected upfront, and I have a model that can predict the concentration profile accurately, and I'm going to now use that to perform the simulations, I should be able to compare the test versus reference. So I don't think the modeling exercise has to be made unduly complex here.
Yeah. So so Joga, I think I get your point. So some of your comments actually tie into the next question. It really depends on how you actually do the model integrated evidence approach. So maybe we can come to our second question. So we probably can tie some of the model validation strategy also together with the second question, I think that would be a little easier. So Donna, yes. So the second question tie into the different designs. So what are the pros and cons of two main types of model integrated evidence approaches? So one approach we have seen people propose in the past is the virtual bioequivalence. That model is built on a small sample set. It is used to simulate the results of a larger population. So in other words, you build a model on a smaller study and you validate your model. You are confident your model characterizes the different level of variability. Then you enlarge the study for that to a larger, well-powered study. That's the first approach people have proposed in the past. So another way of alternative approach is you actually do a powered study, and however, you truncate that study. You do a short term. So for example, for the patient steady state PK study, you do not wait until the new steady state. So what you do is you do right after switch, you continue those individual subjects, you do a study right after switch, you stop there, you do not do further study or PK sampling, and you use modeling to continue the in silico dosing to the exactly same group of individuals, and then you predict the PK parameters or PK profile of those individuals. So I think depends on this which approach you are take. Probably the validation requirement maybe will be different, and as I explained, and there are some pros and cons for these two approaches. So I think this might be a better way for us to discuss, tie into the first question and the second question together. So I'm going to pause there, and I'm going to just get some of your thoughts on this.
I think Murray actually kind of summed this up earlier, and this, I think, links to what you were saying, Joga. If you are just trying to continue the profile to the exact same group of individuals, you can build that for your new test product quite easily. In the case where you've got, you build a model based on a small sample size and then use that to simulate for a larger population, the question then comes is, okay, Joga, you said that you don't need a population PK model, but in order to build...
I didn't say that. I said you still need you to build a population PK model, but I think you have been harping on a requirement that there has to be a published PK model for the brand. That is what I'm saying. You don't need...
I wasn't harping on saying there has to be a published model for the brand. What I was saying is if there's a model publicly available, you can
Why
use
do
that
you
as
need
a
a
starting
publicly
point.
available model? I mean, that's published, right? So why do you need it?
I think Andrew answered this earlier.
No,
Andrew
I'm
said
asking
that
you because I don't understand. You can answer, but I don't understand why that's a requirement.
So I think Andrew answered the question, and so I will just borrow his answer.
Please.
His answer was, in the case that you do not have that model, you build that model from the data that you have. In any case, you have to get hold of a model. Either you inherit the model from the product, if it's reference product, if it's available, or you have to build it. So my question at this stage is that if you're having to build that model on a small sample size and then simulate that for a larger population, you have to, if it's only built on a small sample size, you may not necessarily pick up all of the different covariates that may affect your model for a large population. So the idea of building it only has a limited scope of where it can be used.
Covariates have no place here, Parmesh. This is bioequivalence. We don't need covariates. Why do you need covariates?
Covariates tell you how your
I
profiles
understand
are
that, but
changing.
you don't need it. That's not the question here.
The question
Yoga.
is, how does the model use to simulate results for a larger population?
Correct.
So
I agree with
variants
that.
tell you how your profile varies by population characteristics.
So,
It
Yoga,
tells you how
may
the
I
different
chime in?
parts of your PK profile vary according to your sex, your gender, your age, your creatinine clearance, different parameters that vary in a population. And so if you're going to extend that model to the wider population,
You will not be recruiting renal impaired subjects in a bioequivalence study, an abbreviated bioequivalence study, right?
Yoga, may
Sorry.
I chime in here?
Yeah, please.
Yeah, sorry to interrupt. Because I think one important aspect I did not elaborate before the question is a lot of long-acting injectable products, for safety reasons, we are doing the B study in patients. So it's not as homogeneous as the healthy subject PK studies. So
Yeah.
that's where the covariates may be able to helpful to explain the PK difference. So just to chime in for that.
No, I appreciate that, but my point is that you still try to make your population as less heterogeneous as possible. You're not going to bring in, you said creatinine clearance. You're not going to bring in moderate renal impaired, mild rate. You will exclude them from such studies. You have to come up with a narrow set of inclusion exclusion criteria. It's just bad idea. Even if you don't use modeling, you don't want to do that.
I agree with you in that regard. Ideally, you want to use a homogeneous population that is most sensitive to detect formulation difference, no doubt about it. But I don't know whether from recruitment perspective, how much we can really control the body weight or everything else. That part, I think that's probably debatable.
Yeah, body weight is given, and that's we all know how to handle body weight as a covariate, but besides the point, I don't think it's practical, Parmesh. Parmesh, it's practical to aim to rebuild a population PK model that the brand has built. It's an undue expectation on the generic company. Not possible.
I think sometimes it was. Yeah, go ahead, please.
If I may come close on this topic and then get back to the discussion, and I think Joga's comment do bear merit. Like for population PK, you do not need a pre-existing model. I think you can always develop one, and also you may want to, if there's no covariate by formulation interaction, then there may not be a need to figure out the covariate effect on PK. Mainly we are detecting the formulation performance difference. In that regard, if there's a covariate by formulation interaction, yes, we may want to figure that out. But otherwise, I do not see there's a need. I see Andy's raising hand.
But I was going to change. I was going to try and answer question 2 instead, if that's okay.
Yes, please.
Because I was thinking about the difference between simulating a population of individuals or simulating the same individuals from a smaller study. And I guess in the smaller study example, then you simulate out data, and then you're going to be using the NCA approach, right? You're going to be using non-compartmental analysis approaches. So maybe the difference between 1 and 2 is that 1 is going to be using a fully model-based calculation of areas under the curve in Cmax, whereas number 2 would be NCA-based, and that NCA is known to be a more problematic, especially if you have sparser data or if you have variability that's different at high concentrations than low and things like that. So I wonder if the difference might be there. And then I was also thinking that in the in silico approach, I'm not sure if there's high shrinkage in your model when you simulate out to new situations that you might end up with shrinkage problems, issues with shrinkage if that's a problem in maybe you have low data per individual. So I wonder if it's sort of situation dependent, which one you might want to use.
Andrew, I have a technical question back to you. Say if we want to have like a bottom line small sample size to validate the model, to enable the model to qualify the model to conduct the BE trials studies. From shrinkage perspective, we do have a calculation in terms of a sample size, right? To have a sufficient characterization of the uncertainty. So in that regard, what's your evaluation, for long-acting injectable for the typical ones? What the sample size needed to address the shrinkage problem?
I mean, it's what shrinkage is going to be dependent on
the PK sampling as well. I guess if you have rich PK
sampling, and it's also, of course, dependent on the number of ADAs, the number of random effects you have in your model.
Yeah.
And the number of individuals. So I think it's very dependent, but I suspect most of the problem is the number of observations per individual in terms of shrinkage.
Yes, I agree. It concerned me a little bit now, thinking if we can't, because we are doing dedicated PK study anyway, we can easily circumvent the sparse sampling problem by having more rich sampling schedule, right? The shrinkage may not be a blocker in this way.
Exactly, if you take rich samples. And plus, if you take your model, once you fit your model to data, then your model should incorporate the uncertainties so that you understand the uncertainties, which is very related to shrinkage. But making new simulations of individuals won't necessarily be shrinkage dependent. It will just be more uncertain if you have low numbers of observations per individual. So I think the simulation from fitting to data is less problematic than extrapolating if you have high shrinkage in individuals.
In the approach that we have tried, so where we fit the reference data, single dose, and then fit the model to the data to get the individual parameters, and then extrapolate to get the carryover, we have tried to use the EBEs, and these were leading to a bias. We could clearly not properly control the type 1 error in this case. And then we have tried using what we call the mean of the conditional distribution. So the conditional distribution represents the uncertainty of the individual parameters. The EBEs is the most probable value from this distribution. And the mean is a mean of the distribution, which can be different from the EBEs if the distribution is not symmetric. And when using the mean from the conditional distribution, in that case, we were able to properly control the type 1 error.
Nice.
So it is indeed a point to consider. And even the way to calculate the mean of the conditional distribution had an impact. So to calculate it, we use a Markov chain Monte Carlo approach, from which we need to sample from a proposal distribution. And depending which proposal distribution we were using, we would manage to properly control type 1 error or not. So indeed, it is something that is a bit tricky and needs to be done with care and really checking that we manage to control type 1 error.
Interesting. Were you using? Did you think about using this idea from Benjamin Ribba and Marc Lavier of sampling within that distribution as well?
Exactly, that was what we were doing. Yes.
Oh, okay, nice.
I'm curious, both of you. So, is this shrinkage leads to bias, or is it really shrinkage is unbiased?
So shrinkage is when individual data is not rich enough to get the true individual parameters, and instead we shrink back to the population parameter value. We have pretty dense data, so shrinkage was really small. It was hard to see by eye on the individual data, but when we work on like many simulated individuals, really trying on a lot of data, even this small shrinkage was leading to a bias. The bias is really then in the fact that either you overpredict or you underpredict the carryover concentration. If sometimes you are above and sometimes you are below, it's okay. It's kind of a noise, and it will more or less add noise in your or lower the power, but it's okay. But if you always underpredict a little or always overpredict your carryover concentration, then you get a bias, and this you will see it in when calculating the type 1 error.
Interesting. I'll have to think about that. I didn't think shrinkage introduces bias. Maybe there's an interaction between the design and the shrinkage. So design can be tweaked so we don't have bias.
You can certainly tweak the design so you have less shrinkage. So,
no,
right?
I'm saying
Yeah.
even if there is shrinkage, I did not think it'll introduce bias. It's less, the spread of the data is narrower, but the central tendency is not altered, correct?
Yeah, I agree that it's not completely intuitive why we have a bias. I would have expected that we are sometimes above, sometimes below, but the central tendency is the same. But
so maybe the design
experimentally,
interaction, we'll have to see if there are some design protections can be built.
And I think we don't always see uniform shrinkage. Sometimes you'll see shrinkage on one side more than the other side, or the other way around.
Yes, this could explain that it leads to a bias.
Cool.
Mm-hmm. Interesting.
Can I ask you guys one question? This is now we are, we're trying to find the utility of this method. I really like the idea that that we coming up with a reasonably small study. I don't say it's a small study. It's a reasonable study that where you have some strength in it. Andrew, that the BE studies are always very sampling intensive. These are not, these are not population PK studies. These are intense, BE studies. I will say anywhere from 14 to 20 samples in a profile minimum. That is kind of thing that I see all the time, anywhere from 13 to 20 samples. So the model specification is something not really that possible here, but one can say always I see that power is not there. So in these pilot studies as well as the studies that are abbreviated that we talked about, so you do the study and the study is really good and you can actually model it, but the thing is you just don't have enough power to declare the bioequivalence. That's the most of the time the problem. So what do we do, to say that the model is, we can say that we have a model from the abbreviated data and then say, now I can confirm the bioequivalence. What do you guys say? Like, what should we do there? What are the things that we need in that abbreviated study and when we develop the model there? And then what do you want to see before we go into the final simulation?
So we actually did some research in the past with Uppsala University, trying to address the question you are asking just now. So I would like to turn to Andy. So can you give a really high level response in terms of like how small the study can be, we have enough confidence in terms of that study is good enough to really to build a model for that model to be qualified for the MIE approach for the LAI product?
I'm not sure I can answer how small the study can be, but you can certainly reduce the study dramatically with a model-based approach compared to a standard non-compartmental analysis approach. What you need is a model that can estimate the differences between the two treatment and reference, or sorry, the reference and test product, and then of course you need the uncertainty, and you need to estimate the uncertainty accurately. And so we've spent a lot of time in the last few years trying to figure out how to best estimate model and parameter uncertainty. We think that using a model averaging approach, so potentially using multiple models, especially if you maybe have a complicated model from your from some maybe a publication. In the case of paliperidone, you might have that, for example. But then you're going to have to reduce that model if you're running just a standard BE study in any with the data you'll get from that BE study. And then you need to figure out how to reduce it. So you have a number of possible models. And then you have to figure out for each of those models, how do you determine the uncertainty? And I think this approaches like what Géraldine was talking about, looking at the posterior distribution of individual parameter values, that gives you a non-symmetric uncertainty. You can do the same thing with something called a sampling importance resampling to get uncertainties that are the correct distribution of uncertainties of your parameters. But then figuring out how many individuals you need it's very specific for different situations. But Lucy, maybe you were thinking of something else that we did that I didn't remember.
No, I think you touched some very important elements. I think what you might recollecting, I think some of the access simulation, it appears to me like given the complexity of the long-acting injectable product in the PK model, actually we may not reduce the sample set to a level we really hope to reduce. I think that's what I remember. So I think that's the challenge we are facing right now, right?
Yeah, so we were seeing with these switch studies that there was a significant increase in the number of individuals compared to a steady state study where these very long steady state studies. So you certainly have higher numbers of individuals, and then the question is how high is high, and is it better to have more individuals that sit in a study for 3 months, or is it better to have a few individuals that run 2 years in a study?
Right, that's exactly we are trying to find the relative better approach between this approach 1 and approach 2.
Yeah.
So Murray, early on, I see you were trying to give a comment. Can I come to you?
Yeah, I mean, all the comments that we make are really dependent if we choose number 1 or number 2 here. It's a completely different ball game. It's a completely different research project, if I can say. But in terms of sample size, the number 2 is not associated with a sample size that is that big if you can replicate test and reference in your pivotal study design that you're going to use to establish BE. So if you can use the scaled average bioequivalence approach, you only need to finish with n equal 24 or 28. And that's if it's highly variable. If it's less than highly variable, then you're fine. And regarding the number 1, which from a theoretical point of view is very appealing, that you would have a small sample size and that you could estimate what would happen with a large sample size. We've done some simulations in-house here, and when we do that, it's actually analogous to extending the confidence interval limits. So, for example, if let's say you estimate that you need, 100 subjects to pass at 80 to 125, and you want to, and you want to do that using a study with 30, and then simulating 100 later on, then it's analogous to extending the confidence interval that the n equal 30 gives you. So the central tendency is fine, but the problem is on the confidence interval limits. So, but that's based on the simulations we've done in-house. I mean, I welcome other people working on it, but that is the problem. So far, the Office of Generic Drugs has not been keen on enlarging confidence interval, except in very rare circumstances. So, so that's where it's appealing, but I don't know.
So Lucy, I think one way to come up with this sample size for the abbreviated study is going to be to you'll have to start with an understanding of the expected model. There's no question about that. Okay, it's not saying you have to have a published Popper model. That's a different topic. So you have a idea about the reference profile. Generic companies must have that. Then you say that based upon label or wherever you get estimates of the variability, then you say if you do simulations to plan for this abbreviated study, then you say what would be a sample size that can estimate a true T/R ratio of, shall I say, 0.8 with some precision. I don't want to pick a number that becomes then the new norm, but you want that to be with certain certainty, but that 0.8, then that sample size is the one that you would want to go forward with. So if you imagine a plot with the 0.8 and then the confidence intervals around that, and the x-axis is different sample sizes, as you increase that precision will get better. We can have some kind of a expectation saying it should be at least some number, and that is how you would get the sample size. Now that number can be also derived, making sure that it doesn't, meaning wherever you get the minimum number of minimum sample size, that does not compromise your type 1 error. So that sample size, so the sponsor can produce these simulations for their product and submit to the FDA for your consideration. That would be, to me, the most objective way to go about this.
Yoga, I totally agree. I just want to put a little plug in for, you can also do optimal design to do a similar type of calculation. If you want to first explore the space and then do some simulations at specific sample sizes.
Sure.
Thank you, Joga. I agree with you. I think that can be one way to look at this problem. Okay, I think we had a lot of intense discussion on question 1 and question 2, so maybe we can move to question 3 now. We only have a few minutes left. So we touched upon the approach, so now it's really coming down to the study design. So what are the other innovative study design that can control the type 1 error and also maintain the study power with the most efficient sample size study duration? So the thinking behind is right now that you say that design people are trying to be a little bit in so-called more efficient. For example, we really see some people do repeated measurement as statistic. So that helps stabilize the model, give you more information, and also people actually test the different sequence, different designs. So we want to hear from you that in addition to notes, I mean, we provide the background document to you guys. In the background document, we give you some kind of that what we have seen in the past, such as sequential designs. In addition to all notes, we want to hear from the panel members what are some other innovative study designs?
Well, I presented some earlier. I mean, obviously very quickly because of the timing, the duration of the presentation. But I think if for the model base to give you really all the power that you need to have a crossover design. So I think what other people like Andrew and other have called the switch. I mean, there's not one study design. I mean, it's so I think you're going to have to show ahead of time before doing your innovative study design, through modeling and simulation. You're going to have to show that the design that you want to do controls the type 1 error, maintains the power. Having more power is a problem, okay? Because theoretically, having more power, that should give you more type 1 error. So I just want to make this distinction here. Yeah, so, but this has to be proven ahead of time, and there's not going to be one study design for everything because those long-acting injectable products are not exactly the same between them. There's some differences. Yeah.
Marie, I have a follow-up question for you regarding study design. Should we follow a guiding principle on whatever design you're going to take? You would like to dose both test and reference on the same individual? Or
Yes, because because that's a crossover. Yeah, absolutely. Because otherwise, that's where you need covariates. You know, like before we talked about covariates. No, we don't need covariates, and yes, we need covariates. If you have a crossover design and you're doing the situation 2 from question 2, right? You're continuing people. You don't need covariates because you're going to have individual results. You're going to have the true results per patient.
Yeah,
But if you have a parallel, then it complicates things and it's less robust, much less robust.
Thank you.
Sorry.
You know, that's my notion too. I just want to see if the panel have any difference in opinion for that.
Well, you know, obviously, I like crossover design, like in typical bioequivalent study, because we do sample size tremendous. But I thought with LAI, you know, using truly crossover design is problematic because of the long washout period. So yeah, it's easy to say that we prefer crossover design, but you know, we need to tackle the issues of long washout, right? I know some have defended the use of modeling to simulate a carryover of drug from period to period 2. But it requires, again, modeling, and I myself like to go with perhaps a more simplified approach where we don't need as much modeling as possible. Simply because it just, as I said earlier, a lot of people perhaps don't understand modeling a lot, and a lot of them they don't trust modeling too. So you know, the less modeling we use, perhaps is more easily accepted by other regulatory agencies or even by clinicians.
Yeah, with that, I do see a very important question from the audience. You know, that reflects one of the challenging nature of long-acting injectable. For crossover study designs, if we want to dose the individual with 2 products, certainly we are going to not to have a complete washout. The question is that whether, you know, can we use the example of the model without complete washout, for example, of the drugs with the long half-lives? Would FDA be accepting this approach? And the follow-up, more like a follow-up question, if the model supports, do you need a washout period? And I think that's very, very good question. My opinion is that we should be open-minded, but there should be sufficient justification. There would be sufficient control of type 1 error, and also certainly I think that study design probably can you power to pass B, and but this is not a pre-meeting anything. We can only talk about the general guidance, and if you have a solid approach, you're welcome to submit the package to discuss with us.
Certainly. I think we pretty much ran out of our time for the panel discussion. And I want to thank you all, the panel members, for the active engagement and sharing your inputs, and also for the audience for your engagement. I saw a lot of questions that for the interest of time, we did not get to address all of your questions. And we will, after this workshop, we actually will put together a white paper and to summarize what we discussed, and some of the questions put in the chat box, we will try to address in the white paper. So please stay tuned.
See, Liang Zhao, Liang's there. I think everyone knows who Liang is. He's part of the panel in part. He's Director of Division of Quantitative Methods and Modeling in OGD. He has a broad spectrum of scientific management expertise from industry and the regulatory agency. Liang, can you provide some final comments?
Yeah, thank you, Jim. First of all, I just want to thank the CRCG to make this great event happen. And, without your help and support, there are many things will become impossible. Gather all the stakeholders and the relevant audience to allow a very productive discussion. I also want to thanks to all the presenters, and you can appreciate how much work is behind your presentation. I think that all the presentation are of high quality, in my opinion, based on my past experience of attending, some of other type of workshops, and I really appreciate the effort and thought put behind all the presentations. I also want to thank to all the panelists to share your expert opinion and your really, really deep insight in the issue from both academic, industrial, and regulatory perspectives. That will allow us to better serve the public health, better serve the agency, and better serve the generic drug industry. I think we have achieved a great deal, but as Lucy mentioned, I feel we can keep discussing for another good 2 hours, and then we there are still leftover issues for us to address. Having said that, I think we cannot address all of them today. We are at the beginning stage of countering the challenges of developing long-acting injectables, and I'm sure we'll see each other again in the future. Certainly for model integrated approaches, it can lead to potential cost savings for reduce the sample size and or reduce the study duration. This certainly represents a common interest among all of us, including industry, regulatory agency, and the academia. Of note, the value proposition should not be done at the expense of generic drug product quality. I think, again, the purpose of the workshop is to bring all the stakeholders together to brainstorm on the best practices of using the model integrated approach in the future. The bottom line is that the best practice is not there yet, but we are all actively pushing for it. The best practice can have several key elements. For example, what will be the most efficient study design to allow a model integrated approach, and what will be the best practice to validate the model. This is more in response to the question I see from the audience and from also Raja, that what we see from the industry. What do you want to see? What do you want to see if we can qualify a model for model integrated approach? Certainly, we have spent a great amount of time discussion how to validate the model. I think once the model is fully validated, that it can allow sufficient control of type 1 error and AU power. That's a good model. And also, another element of the model, another element for the best practice is what will be the associated PK metrics associated with the new study design. I think that's been discussed in one of the presentations, but I think we didn't discuss that in the panel, but we can certainly follow on that. With that, I will say, Lucy mentioned, we'll try our best to summarize the valuable information and the insight we have collected in the workshop. The workshop slides and videos will be made available by CRCG at the CRCG website. It's complexgenerics.org in 3 weeks. Again, I appreciate CRCG's effort in making the sharing possible in 3 weeks. With that, I just want to thank you again for all of your effort. And I'm pretty sure we'll see each other and talk to each other again, either in conferences or in regulatory meetings or in other venues. Thank you very much. Have a great evening.